Hi, everyone, and welcome to Let's Talk Risk, where our goal is to connect, engage, and inspire each other as we build a global community of risk practitioners. I'm your host, Naveen Agarwal. And guys, today I'm super excited to welcome my guest, Marina Daineko, who has deep expertise in biocompatibility assessments. She's currently working as a biocompatibility consultant based in Europe, and she specializes in 93-1. She has done many different regulatory submissions or assessments aligning with 14971 and also MDR. So she has a lot of hands on experience in this area, and I'm hoping that this is going to be an awesome conversation. But as before, guys, you know that this is a very casual conversation and you are a part of this. So please feel free to share your questions and comments in the chat, and I'll bring them up here as much as possible. So with that, Marina, thank you so much for joining. Welcome to the show. How are you today?
Hello, everyone. Happy Friday. Hello, Naveen. Really excited to be here and talk about my favorite topic. It's about biocompatibility. So just can't wait to start talking about it.
So that's great, Marina. The first question I have for you, and I'm personally very curious about. I've heard that 10993-1 is going through an update. I don't know where it is right now, whether it's public information or not, but to the extent you can share with us, can you tell us what we can expect in this updated version?
Absolutely. Actually, right now, the standard is under finalization, meaning no more technical changes are expected, only some grammar and some things related to this. And it was really funny to see, really excited to see, maybe it's a better word, that just in year and a half, people who are working on the standards just agreed about updates, because these updates are pretty interesting and they are deep. What does it mean? For instance, upcoming version of ISO 10003-1 would have even more information and more alignment with ISO 14971 about risk management. It means that we would have to apply even more principles of risk-based approach and consider biocompatibility from this risk perspective even more. And this is pretty challenging. For instance, right now there is even more information that we have to assess medical device through its life cycle. It means not only during design verification activities as it was usually, but also if we are talking about prototype, or maybe we can have some, maybe not biological evaluation as who, but at least some assessment on what is it, what can we expect from the biological safety, from the materials that we are using, as well as in the end of life of medical device, like it's stated, for instance, at UMDR. Also, I'm very excited and I'm looking almost impatiently for another thing, and this is guidance how to use clinical data, meaning post-market information, to justify, to provide additional evidence of biological safety of medical devices. So in a nutshell, I would say this is very interesting how we would have it, and also whether it would be recognized by the FDA. Published.
Yeah. So, Marina, thank you for sharing that update with us. Now, it sounds like interesting and sort of common sense to me, being a risk practitioner, but I hear a lot about risk-based approach. Everyone says we need to apply risk-based approach.
Yes.
Yeah. What does that mean in the context of biocompatibility? Can you share some examples with us?
Oh, I love this question because I believe that this is the most challenging part because, as you said, everyone is talking about risk-based approach, and it means that everyone can interpret it a bit differently, when looking into the biological relation plan and report. I would say that if talking about example and practical application of this principle, so first of all, we have to have the plan. And I believe it's the most common challenge when different companies may reach out to the lab without the plan. And according to ISO 14971, as well as ISO 10003-1, even current version, we have to have a plan about what we are going to assess, how we are going to assess it, like all this information documented and well structured. Also, it means that there are specific biological effects. Right now in current version, it's called endpoints, but in new version, we would have biological effects instead of endpoints. And these biological endpoints have to be assessed. But when we are talking about assessed, it doesn't mean it is not equal to tested, okay? It means that it shall be evaluated. And this evaluation is also kind of tricky thing because here we have to consider different data sheets for raw materials, including packaging. Also consider different existing white papers on materials, maybe some clinical studies existing, also information on competitor devices or other similar clinically established devices. And additionally, if required, if this information is not enough, we may have also some tests regarding this biological effect. So it means that we considering all existing information already, we assess what are the risks to have, like, for instance, I don't know, irritating potential or something like this, and we consider whether this risk is mitigated or not. And if required, do additional testing or even deeper investigation.
Yeah. So, Marina, you mentioned something like endpoints and effects. Now, for people who are really experts in biocompatibility, they may understand this, but many of us don't have much direct experience. Can you just start with the basics and tell us what the difference is and why it matters now that the standard is emphasizing effects, not endpoints?
Okay, that's an interesting question, actually, because look, endpoints and biological effects from current and updated version mean similar thing, but a bit different. What I mean, for instance, if we are talking about irritation and sensitization, I love this example. I published a post regarding it, how to differentiate and what is it actually. When we have allergy, for instance, I applied a tissue mask to my face and I have got red skin when I get rid of it. I had this allergy and it was almost immediate, so I have got irritation. But for instance, when I was working in laboratory with nickel solution, with time, I developed the allergy to nickel because of cumulative effect. So I experienced sensitization, and both irritation and sensitization are part of biological evaluation, because here we shall explore the cell's reaction, or it's better to say maybe our body's response to different chemicals that can go from the medical device to our body. And it's not only about cytotoxicity, for instance, irritation, sensitization. These three actually are called the big three. Why? Because they have to be evaluated for each medical device. For instance, medical device that has contact just with intact skin, or implant, that is like more risky device. But besides this, the biological effects to our body, we have to consider the chemicals or medical devices that can be used for long time, like for instance, different implants. Why it's important? Because it's about systemic toxicity, because there might be some effects that can appear just within the time. Together with Angelina, we made a really amazing series of posts dedicated to this topic. And I really like the example from the Bleeding Edge documentary with a hip implant, when it was composed of cobalt, and with the time, cobalt leached to the body and it caused different effects. And these different effects, believe me, they were very harmful. They touch different nervous systems within the body, and we have to have lessons learned. We have to prevent such things in future. That's why it's very important to consider systemic toxicity as well. Right now, in this new updated ISO 10931, there is even more information about it and more focus on it.
Great. So, in simple terms, can we say that we need to understand, anticipate, mitigate, control the effects of potential non-biocompatibility? Is that like a simple way to understand this?
Did I get you right that you are asking me whether it's possible to identify this non-biocompatible thing?
No, what I meant to say that when we talk about effects versus endpoints, I'm still trying to figure that nuanced difference. And based on what you describe, it seems to me that we are talking about what might happen if there is non-biocompatibility. right?
Yes, exactly. It
Okay,
depends on the medical device categorization because
correct.
not to whole all medical devices shall be assessed like
Correct.
the same.
This is one of those things that you have to understand this in a little bit different way, because we actually want biocompatibility. We don't want non-biocompatibility. So
Absolutely.
from a risk management point of view, the hazard really is non-biocompatible material, not necessary. So sometimes it can get a little bit confusing, but we have to pay attention to what we are talking about. Okay, so guys, if you're just joining us now, my guest today is Marina Daineko, who is an expert in biocompatibility evaluations and assessments for medical devices. And we're talking about some of the common challenges and maybe best practices. But please feel free to share your own questions, comments. And I think Ming, you have already shared a question here. With the ongoing reduction in force at the FDA, do you anticipate a reversal back to a test checkbox mindset, while the FDA is still held to MDUFA timeline commitment? I know, Marina, you're in Europe, so I don't know to the extent that you can answer this. I
I
invite you.
can.
Go ahead.
I'm also doing quite. Hi, Ming. First of all, nice to see you here. So yes, I have experience working with the AFD and addressing their questions. And I would say that, for instance, guidance related to biocompatibility applications, it was updated back in September 2020. It already says about 3Rs approach that stands for reduce, reuse, and third one. All the time I'm forgetting it.
Okay, that's all right. We'll
So
find it.
it means that we have to reuse the data. And additionally to this FDA guidance, there is very nice also appendix that is applicable for the medical device that has contact with intact skin, and it can be provided just simple justification regarding it, and no additional testing is required. So I don't think that considering all the things as well as changing current state of the art, how biological evaluation is performed and is done, that it is not necessary to perform tests just for test. It's necessary to evaluate the things, what we have, what we already have. And additionally, I saw that there is kind of new or not very new concept about real world data and real world evidence. And there is more and more publications to this topic. So I believe we would not get back to the checkbox approach, rather to deeper understanding what is going on with our medical device when it's applied to.
So, but Ming, I think you're asking a very relevant question here. And there's a general concern that the rigor and the work that goes on into doing regulatory reviews, may be compromised. I totally understand where you are coming from. What I've heard from people, and I think Angelina is also sharing that, is I don't think the rigor will be compromised. There might be delays, but the rigor will not be compromised. At the same time, I think, Marina, you are making a very important point. I think it's up to us. FDA has already told us that they favor a least burdensome approach. So it's up to us in the industry to make sure that our assessment is not check the box. We are not relying on FDA to give us a stamp of approval. We are showing them what we think is the best thing to do, not over test it, not under test it, and giving them our case and having them review and give us feedback. So, in fact, I had this conversation with Michael Nilo recently, who recommended that in the light of what's happening with the FDA, industry has to take a little bit more robust, rigorous, risk-based approach throughout the life cycle. And we have to get better. So I think the onus is on us as well. So let's continue. I want to pick on this because Roger has shared this comment. I have always been confused by the use of the word endpoints in biocompatibility. So let's talk a little bit more about that. Marina, can you share an example of what we would mean by an endpoint?
Endpoints, according to current version of ISO 10931, means, like I already said, cytotoxicity or irritation or sensitization. It's actually, if talking like in normal language of healthy person, it's biological effect, but it's called like endpoint because it shall be addressed, because it's language of the standards that was applied in 2018.
Okay. So what I'm hearing you say that right now the standard uses a language endpoint, which actually reflects what might happen as a consequence of non-biocompatibility, and that it's our job is to make sure that we address it through our risk mitigations and testing and provide that evidence. Did I kind of package it in a very simple way correctly?
Yes, that's correctly. But I believe here is in current version, there is even more emphasis, like this is endpoint, and we don't see this is like biological response or biological effect. I mean, for people who just joined this domain, it's not so obvious.
Not so obvious. Yes.
Yeah.
I can see it's confusing. And I think for now, I will table this conversation, but I do feel a need to kind of dig deeper into this, Marina. Maybe at a future time, we can actually go a little bit deeper and use some examples. I think that would be interesting. So I want to continue. Marina, can you share with us some common challenges that people face when they are trying to do biological assessments?
Yeah,
Biocompatibility assessments.
Absolutely. So first of all, it's know your device. I really repeating it as mantra. Why? Because actually all the information regarding medical device shall be considered here. And it's not only Roger. Absolutely. I have the same feeling as you. Sorry for commenting on it. So because all the information, not only like data sheets related to raw materials, packaging, all these things, but also manufacturing additives, even like for instance, what wipes are used to clean the manufacturing lines. Okay, so all these things shall be assessed by the person who is fulfilling the biological evaluation. Second, I would say like the most challenging part here is to tell the story, because we have to describe the biological evaluation for the person who may not be aware about our medical device like so well as we are. So to tell the story, to make it smoothly, to connect the dots, to connect what we are saying in our literature review, how it's connected to our medical device. So everything shall be like a story. You read, you don't have additional questions, at least critical, hopefully, that kind of thing.
Well, one of the things that you mentioned just now, and I don't want to lose sight of that, is what you're describing to me seems like it presents a need to connect our design FMEA with process FMEA. You mentioned something like you might be doing something during the processing steps, wiping tables, cleaning tables, and those residues might be transferred somehow to your device. So that wouldn't necessarily appear in the design FMEA. It could appear in the process FMEA. So I think we need to connect the two. Does that make sense?
Yes, it makes sense. And I also had a similar question from the regulatory body to one of the project I was supporting, that manufacturing line was used for different medical devices, and they were asking about cross-contamination.
Yes.
And we had to describe all the processes, cleaning, etc., to justify that there is a low risk of cross-contamination, because you remember always there is risk. It's also it's just a question what.
And I can tell you how it shows up and presents a challenge from a risk management point of view. This shows up in lack of traceability to hazards, because you have identified some hazards and hazardous situations resulting because of processing issues in PFMEA and some in DFMEA, and they don't talk to each other. And we cannot connect the two and bring them up into a single place for traceability. And that is one problem I have seen in practice. I think biocompatibility assessment presents a very unique case study to us to figure out how to connect the two. So can you, does that make sense to you?
Yeah, absolutely. From ISO 4971 about risk management, this risk exercise, during hazards and these sorts of those situations shall be done by a cross-functional team. It means that not only like myself shall brainstorm on this topic, but I have to set up a team and we have to talk to each other to identify.
So, Marina, we now have another question from Dr. Poonam Meena. She's asking, could you talk about the cumulative contact exposure through the revision, though the revision provides guidance around it, but still it seems to be confusing for some device manufacturers, as it might trigger additional testing due to recategorization. Now, this just went over my head because I'm not in this area. So, Marina, can you just take it step by step and walk us through this question?
Absolutely. It's really very relevant question. First of all, this new version of ISO 10993-1 reconsider the cumulative contact, as well as even right now I can see that regulatory bodies are paying even more attention to this topic. So cumulative effect, first of all, we have to consider it. For instance, if the device can be used like more than once, we have to calculate. We have to have the impression, and usually I'm reaching out to clinical affairs guys who can help me to figure out how this device can be used, how long it can be used, and all the things. And for instance, let's imagine there, for instance, transfusion set or infusion set, doesn't matter. So you may see that, okay, they can be used for like, I don't know, up to 7 days. But this up to 7 days can be used like several times. It means that overall, overall cumulative exposure can go even to long-term exposure.
Yeah.
For instance, let's have a look into other like transitory contact. By the way, EU MDR is saying about transitory contact like 1 hour, while ISA 10931, as well as like practice that is applicable within the industry, is less than just 1 minute. Okay, so if we are considering the device that is used like for, I don't know, 5-10 minutes, like syringes, maybe with a needle, okay? But this needle can be used during treatment like several times per day, and during a week or even 2 weeks for 5-10 minutes, 1-3 minutes even, okay? So we have to consider it even not like limited, less than 24 hours, but even longer. So it will go into prolonged categorization. Why? Because this 1-3 minutes for, I don't know, like 2, 3, 4 times per day and during 7 days. So what I'm trying to say is that one usage of such devices several times per day will go into, will be counted like 1 day.
Yeah.
But if it's like more than 1 day is used, then we go to prolonged exposure. And it's really challenging. And I saw and I help in my customers to categorize it correctly. Yes.
Perfect. So I think that's a great point, guys. Please reach out to Marina for more detailed conversations. But it makes sense to me, Marina. Common sense tells me, this concept is very common in the chemical industry, for example, as far as exposure to harmful chemicals is concerned. And I have a lot of experience in the contact lens business, where you use a contact lens on a daily basis and you take a fresh one out. Over a period of 5, 10, 15, 20 years, you're using these devices and you are exposing your system to even like parts per million or parts per billion level residues or residual chemicals. So this makes a lot of common sense. And I think this is where, when you do risk analysis, you have to involve people who have expertise in biocompatibility assessments and clinical evaluations, your clinical folks, animal studies who have done. And basically, you need to kind of have this information up front when you are developing your design inputs, for example, right? So this makes common sense to me. Great. Thank you for sharing that, Marina. Now, Mark is sharing with us this. I think he has a question. What is industry doing to record and report environmental controls data other than paper-based forms so that data analytics tells you that you have an issue, trend, or a potential for problems with the facility and clean rooms? And in the context of biocompatibility, it could be maybe exposure of the device while it's being processed to these contaminants, and they may be carried through, right?
Yes.
So can you share any best practices in the industry for environmental monitoring that you might be aware of?
That's a really cool question. I saw that there are different programs, applications, that can collect all this information automatically, and they are comparing it to the set up criteria because as per ISO 13485, the environment shall not only be monitored but also controlled. And when we are talking about controlling, it means it shall be changed, and it is adjusted according to the setup temperature, humidity, and some other parameters regarding it. If we are talking about, if it's more related to data analytics, I would say, because surely I saw such applications with like setup parameters, like analyzing online and adjusting them if required. But regarding data analytics for biocompatibility, I would say it can relate not only to environment control, but for analyzing the post-market surveillance information. Because here we have also to analyze the trends and understand what's going on here, because we might have some complaints related to biological safety, and we can go through risk management process and identify risk as low. But if we can see trends, then we have to do something with it to enhance the situation.
I think this is such an excellent point, and it got me thinking a little bit here. And again, I want to emphasize the importance of connecting design FMEA with process FMEA. So, you might have alert limits, control limits in your environmental controls, fine, great. and you might be taking actions if you have non-conformances. But I don't think you can evaluate the impact of those non-conformances. Here, I'm talking about contamination. I'm talking about contaminating the product that could have a biocompatibility effect, right? That kind of biocompatibility issue. Unless people understand this connection between the design aspect of it and the process aspect of it, those NCs cannot be assessed appropriately for risk, right? So I want us to kind of, I think maybe this theme we wanna discuss in the future as well. But Marina, as always, time flies, and we are running short on time. Yusuf has a question, so I'm not gonna say no to that. So let's review it very quickly. The question here is a long. Are there specific challenges or considerations that manufacturers should prioritize to ensure compliance and patient safety? So that's like a very broad question, but essentially in the context of how do you foresee this integration of advanced materials, nanomaterials, and whatnot. So I think briefly I will address this, Yusuf. We can take that on in a more detailed conversation, but this to me is exactly where risk-based approach is so critical in the design phase. You have to look at it, right? Marina, you were going to say something. Go ahead.
Yeah, absolutely. I totally agree with you. And just to be short, yes, there are specific challenges related to nanomaterials because it's something new, and we have to analyze the effects of these nanomaterials from different perspectives, also including systemic toxicity because of lessons learned. And I know that there is a specific group of people who are working on the standard regarding biological evaluation of nanomaterials to detail how they shall be assessed to ensure that medical devices are safe.
Okay, got you. So it should be brought into an early conversation during the design phase. Great. Thank you so much. We are continuing to get questions, but guys, we have run out of time, and I want to be respectful of your time. But it seems to me that there is a need for a more detailed, longer conversation here, Marina. So I don't want to put you on the spot, but I'm sensing from our audience that there's a need to go deeper into it. Would you be willing to come back?
Yes, absolutely. And if you don't mind sharing with me these questions, I would address them and
Yes.
Because I can see like Roger made really relevant comment regarding clean room and bioburden and some other testing shall be done for each batch. It's like additional evidence that all okay here, and there are so many aspects that we can
Excellent.
discuss.
Excellent.
Yes.
Great. So guys, I'm going to give Marina just a couple of minutes to think about a few key bullet points that we can take away after this conversation. But here are some announcements for you. First, I want to invite you to come next week. If you're trying to set up a community of practice in your organization to bring people together, I will have a guest who will share some best practices of having done this in a large organization. How do you bring people together in a company to learn together, collaborate together, and talk about these topics? So please show up. We have these conversations every Friday at 11 Eastern. The second announcement, in case you are not already aware, is the next post-market surveillance workshop that I'm offering. It's a 4-hour workshop, is going to happen on April 9th. I do have a few spots available. So if you want to attend, please reach out to me and I will share the registration information. It's a very detailed, hands-on workshop. It takes 4 hours and we go through a lot of different topics. So if you're interested, let me know. With that, Marina, I welcome you to just share some 2 or 3 key takeaway points.
Yeah. Thank you, Naveen, for this opportunity. So first of all, know your device. Secondly, prepare your biological evaluation plan. And third, apply risk-based approach, but think what does it mean actually and stick to the standard. If you have any additional questions or anything you would like to talk about, just please feel free to reach out to me on LinkedIn. I would be more than happy to talk with you regarding Biocomp. And Naveen, I hope that we can proceed talking about biocompatibility at Let's Talk, please.
Yes. Thank you so much, Marina, for giving us your time and sharing these insights with us. Thank you, everybody, for showing up again and actively participating in this discussion. I wish you all the best. Have a good weekend, and we will see you once again next week. Take care.
Thank you. Enjoy Friday.